Hello, my name is Cheyenne Rosenbaum, and today I will be defending essential oil sensi-disc reaction zones on select fish bacterial pathogens compared to FDA-approved sensi-disc reactions as a thesis topic for my American Public University capstone proposal class, EVSP 601. Before I get started, I would like to mention that you can follow along with my presentation at the bottom of the screen. In order, I will be covering the thesis topic in question, the relevant topics of fish pathogens, FDA drugs, essential oils, and what makes this thesis project significantly different than current research. Again, my name is Cheyenne Rosenbaum, and I am currently working on a Master's of Science in Environmental Policy with a concentration in fisheries and wildlife management. In 1933, the father of conservation and wildlife ecology, Aldo Leopold, stated, "The role of disease in wildlife conservation has been radically underestimated." Leopold, 1933. That statement was true then and still holds true today, which is why I decided to further investigate if 5% essential oil sensi-discs can generate a reaction zone equivalent to or better than the FDA-approved fish drug sensi-discs reaction zones. Related to my degree, I am interested in bacterial disease in a fish management setting. I live in Pennsylvania, which is prideful and dedicated to their freshwater trout populations. One method we use to manage these populations are by stocking from government and private hatcheries. However, as with any high-density population, these hatcheries can easily propagate disease. This is a problem because there are only 8 Food and Drug Administration FDA-approved antibiotics in aquaculture, approved drugs for use in aquaculture 2010, which, with so few antibiotic options, has led to a rise in antibiotic-resistant bacterial pathogens, Lee et al. 2016. The bacteria I will be further investigating are based on trout-related pathogens in Pennsylvania, accessibility of bacterial culture, and best genetic variety. Plenty of fish-related bacterial diseases have overlapping symptoms of inflammation, sores or lesions, and lethargy. In particular, I will be looking at Erysomus salmonsida subspecies salmonsida, which causes the disease frunculosis, commonly known as fin rot. Soprano-embolic 2001. Yersinia bruckeri, which causes enteric red mouth disease, Kummer et al. 2015. Characteristic of hemorrhaging in the mouth region. Enterobacter cloacea, which is particularly antibiotic resistant, causing hemorrhaging, sepsis, and bladder and urinary tract dysfunction, Davin-Regley and pages 2015. Lastly, Adricilia tarda, which primarily affects catfish but can use trout as a host, causing a sepsis disease, Abbott and Jonda 2006. So how do we currently treat these diseases? Currently, one of the methods used is the limited FDA-approved drug sensitivity or diffusion discs. At the Fish Health Center in Lamar, Pennsylvania, we use tetrasilacine 30 micrograms T30, sulfetamine 23.8 micrograms, and oriprim 1.2 micrograms SXT, and fluorophenocal 30 micrograms FFC 30. Here is a typical sensi-disc result on Y. ruckeri. After incubation, these reaction zones form, and based on the diameter, length, and zone clarity, we can speculate which drugs will be the most effective against the specific strains of bacteria. Medicinal properties of plants have been around since the dawning of medicine. In particular, essential oils have been recorded to interact with gram-negative bacteria. Nazaro et al. 2013 and Sadgrove and Jones 2015, which conveniently are the bacteria I have chosen. Also, it is important to note that this is a foundational study. You cannot and should not pour chemicals onto a living animal without good foundational evidence, of which I will create my own 5% essential oil Sensi discs, such as these ones. I have chosen lavender and tea tree oil for its antifungal properties, sweet orange, which is commonly used in natural antibacterial cleaning sprays, and oregano, known to highly interact with bacteria. I also have chosen these essential oils to obtain a representative spread of floral, fruity, and savory oils. Looking closer at these oils and referencing Nazaro et al. 2013, we can speculate how a reaction zone may form in these trials. I will start with lavender and tea tree oil, scientifically referenced as Lavandula and Messina altifolia. The lavender is a fragrant purple flower, and tea tree is a large weeping tree with thin leaves. The active compound in the oil combination is cinnamaldehyde, which is a phenylpropene, simply referring to its chemical structure. How this oil is suspected to work is by disrupting quorum sensing. Quorum sensing is how interbacterial communication occurs, which in this case, we presume it will prevent the bacteria from reproducing. Oregano oil, or oreganum, is from a fragrant, savory herb which is sought after for its leaves. The active chemical in oregano oil is carvacrol, which is a type of terpene. The suspected interaction between the oil and bacteria is for the outer membrane to disintegrate, causing cell death. Lastly, sweet orange, or citrusinus, is primarily derived from the peel of the orange, which is from a fruit tree. The suspected antibacterial compound in this oil is limonene, which is also a type of terpene. The method of antibacterial reaction suspected in this scenario is for the limonene to enter the cell envelope and alter the structure, ultimately causing cell death. The majority of current research on the topic of fish pathogens in essential oils is using essential oil-infused gelatins as a plastic replacer in the seafood industry. Quaison and Coben 2017, Taknaka et al. 2012, and Trexera et al. 2014. I will also statistically compare my results to FDA-approved Sensi disc results. Some research has been completed on essential oils as fish antiparasitics, Travers Diaz and Carnegie 2018, which are pathogens but not bacteria. I will be performing my experiment at safe dilutions. Essential oils can be harmful at 100% concentration, which may be harmful to the fish hosts as well. Lastly, these studies don't look at these oils on these bacteria in a diffusion disc method. Ishmael and Yoshida 2017, Souza et al. 2016, and Souza et al. 2017. This is why it is important to investigate if 5% essential oil Sensi discs generate a reaction zone equivalent to or better than the FDA-approved drug Sensi disc reaction zones. These are my references.